Thanks, Nick, for a nice presentation. And I would like to move to the next speaker, Dr. Spira, Professor of Medicine, in the lead director of the Nottingham Digestive Disease Center Biomedical Research Unit at the University of Nottingham. He was the editor of the Gut from 2003 to 2009. His main interest is in the pathophysiology of functional GI diseases, focusing on infection and inflammation in IBS. He twice edited the British Society of Gastroenterology guidelines for the management of IBS. He's a member of the board of the Roma Foundation. So the title of his presentation today is GI Microbiology, Pathogens, Exposure Patterns, and Immune Responses. Dr. Spira.
Thank you very much. Oh, perfect. Right. So, great pleasure to be here. I want to thank Ami for organising this and all his members of his committee. So, I've been asked to talk about the microbiota, and this is truly a very exciting area. There's been a revolution. So, when I was a medical student, microbes were things that grew on agar plates. The problem about using this technique in the gut is that all the different microbes require very different conditions, and it would take truly a very long time to culture a single individual's organisms. And this has been radically changed by the use of non-culture techniques. So it turns out that there's a gene, the ribosomal RNA gene, which is very well conserved across all bacteria. So you can quite easily generate PCR probes that will produce bits of DNA of this gene, copies of this gene, which differ subtly between bacteria. And what you can do is run them out on a gel, as has been done here, and you get these little bands that correspond to different species. And you can type them, cut out the band and sequence them. And in fact, this has now been sort of superseded now by amazing advances in the technology. So that this device called 454 barcoding pyrosequencing, don't ask me what it means, but what it means is you can get 1 million reads characterizing this gene and describe in great detail every individual's microbial flora. And of course, what have we learned using this very sophisticated technique? Well, we've learned the obvious, that it's very complicated. And if you get this information, you can produce this thing called a dendrogram, where the proximity of each individual bacteria relates to the proportion of similarities of the genome. And you can see that we get big groups. Don't know if this works. Yeah. So we have here in very small print the Bacteroidetes, the gram negatives and the gram positives down here. And we've learned using this technique that it's not only different between individuals, but in the same individual is different between the lumen of the gut and the mucosa. So we've got a distinctive mucosal-associated bacteria. And even within the stool, there's heterogeneity because some bacteria are very intolerant of oxygen and some like different conditions. So here, for example, in the stool, there's a little layer of mucus on the outside, and there you can see these mucotropes, whereas some of the other organisms will be only found in the more anaerobic interior. Now, this microbiome is very complex. It does depend, I'm sure, on the genetics, although it may be difficult to show, because that's likely to alter GI physiology and also the immune system. And there's important interactions between these two systems. So the immune system influences the microbiota, which secretes immunoglobulin, for example, and various defensins. But equally, the microbiota alter the immune system. So if you don't have them, your immune system is very different. And likewise, there's an interaction here between gut transit, for example. So the microbiota are very important in digestion, and they will changing the microbiota will change rates of transit, for example. Now, on top of that, we've got diet, which varies obviously worldwide very substantially. We've got pathogens. Again, there's a very distinctive geographical variation there. And then we've got antibiotic use. Again, differs. So in the short time available, I'm just going to focus on these two, the diet and the immune system. So the important message is that it took us 2 million years to move from the early hominids to modern man. 10,000 years to move from first culture of the first agriculture to modern agriculture, mechanized as we have today. And just 200 years to move from a time when we were all eating basically food we'd grown ourselves to a time when we now eat food that has been grown by somebody else and usually extensively processed. But of course we've still got the genes of the hunter-gatherer. So what difference will this make? Well, we have to answer this question, we have to go back and find a hunter-gatherer. Now, there aren't many left in the world, but in this place, Burkina Faso, which I never knew about until I read this article, a little country in the middle of West Africa, they do have still a very rural environment. I wouldn't say this is hunter-gatherers, this is more medieval, with very little mechanization. And their main diet is sorghum. And this is it. This is what they live on. And if you compare that, in this study, compared with Florentine children, where, of course, there's supermarkets and pizzas, and this is the diet comparison. So the bottom line is the total number of calories in Burkina Faso is just under 1,000, and 3.2% of the diet is fibre. In Florence, the total calories are 50% higher, and the amount of fibre is a much lower percentage. Now, the Burkina Faso children are eating mainly carbohydrate, mostly from millet. They eat a lot of legumes and some vegetables and fruit. In Florentine children, they're eating similar amounts, but they're eating a whole load more of things that they don't have at all in Burkina Faso, which is dairy products, meat, eggs, oil, very little meat in this diet, snacks, and, of course, a higher calorie intake. Now, this makes a huge difference to the microbiota. And here you can see your Florentines have got a lot of the red, the firmicutes, and very little Bacteroidetes, whereas the Burkina Faso are nearly all Bacteroidetes, and they've got some that we're not familiar with. This one, I draw your attention, Xylanobacter. This is called this because it can break down xylans, and xylans are found in sorghum and not much else. So their bugs have adapted to the diet. It's obvious, really. They live on what's left over after it's passed through the small bowel. So much for diet. What about infections? Well, I couldn't find a map of the worldwide distribution of all acute infections, so I've had to go for cholera, which is a map the World Health Authority provide. And I think the infectious diarrheas probably map the same area. So it's basically the tropics, particularly Africa, Asia, and particularly if we focus now on Bangladesh, in this community survey, the average child got 2.5 diarrheal episodes per year, and in the Netherlands, this was 0.02. So 100-fold difference in the incidence of serious infections in childhood. Not only is the incidence different, but the sort of organism is quite different too. So in the Netherlands, viral gastroenteritis is the commonest, followed by bacteria and protozoan. In Bangladesh, virus is very little importance, enormous importance in worms, particularly Trichuris, and protozoan and bacteria as well. So very different organisms. Now, the next couple of slides I owe to Graham Rook, who has promulgated the hygiene hypothesis. So he maintains that there's been a reduction in our exposure to microbiota in our early life, and this is very important for educating our immune system. With the reduction in infectious diarrhea that we've seen in industrialized countries and a decline in worm infestations, there's been a striking rise in allergic disease and a rise in inflammatory bowel disease as well. And this is a map showing a cohort effect of the prevalence of asthma, which really took off in the UK and in Europe in the 70s, and the lines like this. And then here we have countries that traditionally didn't have much of this, who are now acquiring it, particularly in Asia and Japan. And exactly the same map could be drawn for Crohn's and ulcerative colitis. All countries are showing an increase, and there's a lag across as different countries adopt more similar diets. So we need to realize that we have, as Graham Rook calls it, old friends. So we have microbiota that have been with us for millions of years. And it may surprise you to know the lung has got a microbiota. Not many, but there are normally bacteria in your lung. In your skin, there's a lot. 10 to the 7th. In the gut, we know there's a great deal, 10 to the 13th. And then also we have what Graham called pseudo-commensals, things that don't live in our guts but pass through them. So unless you're drinking, if you're drinking ordinary water from a spring or somewhere like that, you'll have a lot of microbacteria in them. Fermented foods, in the past, we used to eat a lot more. They've got lots of lactobacilli. And we were traditionally exposed to helminths in substantial number because, of course, they're found in the soil. And the point about old friends is we've evolved. We have learnt, our immune system has learnt not to make inflammatory responses to these. And, in fact, our immune system depends on them. And the way it depends on them is because these old friends, worms, lice, and mycobacteria, have this property of stimulating antigen-presenting cells to encourage the production of T-regulatory cells. And these cells, in general, produce IL-10, TGF-beta, and they downregulate inflammatory responses. And when you get exposed to a specific antigen from the gut contents or an allergen, then in the presence of these stimulation of these antigen-presenting cells, they're more likely to produce a non-inflammatory response and to become tolerant. So the absence of these friends leads to an over-responsive immune system, and hence allergies, asthma, IBD, and potentially MS. So we pursued this idea then that childhood living conditions would be important in post-infectious IBS, and we've heard some comments about this already. Now, we know that infectious gastroenteritis is very common in underdeveloped world and it relates strongly to childhood living conditions. So the risk factors are known. If you have animals around, if you live in a rural environment, have lots of brothers and sisters, and if you lack running water, you're much more likely to have an infection. But then this is the catch. In many infections, for example, we know a lot about Campylobacter, they're ubiquitous. And in some areas, most children have had 2 or 3 infections by the time they're 3 years old. And the characteristic feature of Campylobacter in childhood is it's quite a mild disease, whereas in adulthood it's quite severe. So we know that the risk of developing post-infectious IBS strongly depends on the severity of the initial illness. And our hypothesis was, therefore, that the risk factors for childhood gastroenteritis would actually decrease the risk of getting severe gastroenteritis in adulthood, and therefore decrease the risk of developing post-infectious IBS. So we used the power of the internet to survey over 1,600 people worldwide. Now, we divided them into North and South. So in Europe, the Alps was the division, and in North America, it was the Rio Grande. And what we looked for a self-report of the following features to diagnose post-infective IBS. So these were people who said that their illness began suddenly, 58%, while travelling, 40%, or with an initial illness characterised by fever, vomiting, bloody diarrhoea, or a positive stool culture. And of our 1,600, 300 met this criteria, an incidence of 18%. And then we looked at the north-south divide, and we found that the northerners, 25% of them, or about a quarter, described post-infectious origin compared with just 12%. So that's an odds ratio of 2. So you're twice as likely to develop post-infectious IBS. So very briefly now, as the orange button is showing, what about the microbiota in IBS? Are they different from normals? Well, I think I've listed here a summary of many studies, and you don't need to read all the things. The point is they don't produce the same conclusions. They find differences, but they're all slightly different. And it may be not that it's specific bacteria that's different. It may be the pattern of them that's different, or their variability. So if we accept that these studies have shown differences in IBS versus controls, what might be the cause of the differences? Well, the first thing is probably us. We give pills to people, and one of the pills we give out in vast numbers, 44% of IBS in the US tertiary care are on a PPI, and that, of course, prevents your gastric acid killing bacteria and allows exposure of your small intestine to abnormal flora. We know that in this survey from the Mayo, about a third of people with IBS have either abnormally fast or abnormally slow transit, and that might well alter gut flora. We know it does, in fact. We've seen recently evidence from that there's a mucosal immune activation in some patients with IBS-D, and that certainly will influence the flora. And, of course, our patients often alter their diet. In particular, they often exclude things which might normally be food for bacteria. So that's likely to change the microbes. And then, of course, we have the phenomenon of the pathogen colonization, which will differ worldwide. And then prescription of antibiotic use. Patients with IBS often have multiple somatic symptoms. They see the doctor more often than normal people, and this may often lead to an antibiotic prescription. So very quickly, dealing with the last two, what effect does acute gastroenteritis have on the gut flora? Well, it reduces short-chain fatty acids and increases the pH, and this changes the ability of some bacteria to grow. In particular, it usually allows an overgrowth of enterococci and facultative anaerobes, which are normally inhibited by the low pH. And it depletes the normal anaerobic flora, and this overgrowth of enterobacteria is known as dysbiosis. So antibiotic use, which is commoner in industrialized countries, aggravates dysbiosis, and we know that it increases the risk of developing post-infective IBS in traveller's diarrhea. It also increases the risk of developing IBS in the UK in this study, which just looked at the incidence after a prescription of antibiotics. So, summary slide now. I hope I've shown you that there's been a huge change in DNA-based techniques for assessing the microbiota, and it hasn't produced any answers, but I certainly think it's opened the door to new understanding. The gut microbiota is substantially influenced by genes, diet, and pathogens, and these differ substantially through the world, so we would expect differences there. And there's been this huge change in our diet associated with agriculture and urbanization that has outstripped the genetic coevolution of commensals and humans and might produce abnormalities. And the hunter-gatherer diet was very high in fibre, low in meat and milk and fat, and this produces a different ratio of firmicutes to bacteroides. We've lost our old friends, some of which we're quite pleased about. Haven't got lice and worms anymore, but this may increase the risk of post-infectious IBS. I just finished by acknowledging the people who work with me, and thanks for listening.